and more than 60 research scholars and students from different colleges and universities are presenting their posters. Now I would like to invite today's honorable chief guest, Mr. Vikram Reddy Garu, to address the participants with his valuable message. Thank you.
Thank you, sir. Good morning, one and all. A great day. We are conducting another international webinar, that too in the very critical field of science today, which is more need of the hour. And we have eminent personalities here joining us: Dr. Challa Suresh Garu, Dr. Srinivas Rao Garu, Dr. Shahnaz Majeed Garu, and we have another Dr. Gulab Khan Rohela Garu, and to the convener, organizing secretary, co-convener, organizing committee, to the chief patron, our chancellor, Dr. CHP Purushottam Reddy Garu, and the guests of honor, and a registrar, Ravinder Garu, to the entire team who has put in their efforts to bring in this webinar that is going to help the students and the faculty. I'd like to wish you all a good luck for this webinar, and also would like to congratulate your efforts in organizing it. So we know how DNA profiling, cloning, or genome analysis, stem cell and tissue engineering are a kind of emerging things on today's date which are helping the mankind. Biotechnology, using the natural resources or using any kind of resources that are available, plant-based resources, we are trying to create an alternative path where we can reach out to more easier and a cheaper way to serve the mankind. Biotechnology has been, from the past 2 decades, contributing in multiple ways to bring very close to the organic living and lifestyle of the human being, and hopefully with all the product applications and all kind of technology, all kind of science that is emerging,
Reddy sir, unmute yourself.
Sorry, sir. Was I audible or?
Yes, sir. It is audible.
Okay. So I'm out somewhere. So I'd like to hand over to Dr. Rajendra sir to introduce the guest speakers or take the program forward. I'm looking forward to some interesting information that is going to come from the emerging personalities and the experienced leaders of this industry. I'd like to wholeheartedly thank them for giving their time today. And I also want to participate along with all the students and the faculty to learn something new on today's date. Thank you so much.
Thank you, sir. Thank you so much for your valuable words, sir. Now I request our registrar of Chaitanya Deemed University, Professor M. Ravinder Garu, to deliver his message.
Good morning. Honorable Chancellor Sir, Dr. CHP Purushottam Reddy Garu, Vice President, Vishwabharati Education Society, E. Vikram Reddy Garu, In charge Vice Chancellor, Professor Krishnamacharya Garu, resource persons, conveners, organizing committee members, warm greetings. At the outset, I cordially welcome today's resource persons, Dr. Challa Suresh Garu, Dr. R. Srinivas Garu, Dr. Shahnaz Garu, and Dr. Gulab Khan, who are going to speak on Alzheimer's role of microbes, cancer treatment, and Biotechnology, respectively. We are glad to have these distinguished scholars at this seminar We have so far conducted 17 seminars on different themes. I'm happy that the Department of Biotechnology organizing an international seminar on new trends in biotechnological applications for human welfare on 26th and 27th November 2021. The theme of the seminar is relevant since it focuses on the challenges and issues in biotechnology to create a research ambience among the students, research scholars, and the faculty members. It is great pleasure that the potential academicians and persons from industry will be taking part in the conference. I am of the opinion that the deliberations in the conference will enable the participant to solve the unknown challenges and provide us better future. I congratulate the staff, research scholars, and students of biotechnology on the eve of organizing the seminar, which provides an opportunity for the fruitful interaction during the deliberations. This helps us to acquire the knowledge and skills on the theme of the seminar. I wish the seminar a grand success. Thank you, one and all. Over to Professor Rajendra Garu.
Thank you, sir. Thank you, Registrar sir. I'm extremely sorry, sir. Now I would like to invite our in charge Vice Chancellor of Chaitanya Deemed to be University, Professor Kishanpura Garu. Please give some valuable message for us. Thank you, sir.
I am audible, sir.
Yes, yes.
Good morning, everyone. Esteemed Chancellors, Sir Purushottam Reddy Garu, respected Vikram Reddy Garu, Registrar Ravindra Garu, and Anuradha Ma'am, Rajendra Garu, other dignitaries of this international seminar. I deem it a privilege to participate in the international seminar on new trends in biotechnological application for human welfare, organized by the Department of Biotechnology, Chaitanya Deemed to be University. And the topic chosen for this international seminar is very contemporarily relevant and very suitable in the present context. Now, I congratulate all the organizers of this seminar for their relentless efforts put in by them to make this international seminar a grand success. I time and again say that organizing seminar is a part of intellectual exercise wherein the latest developments in the chosen field and the knowledge in the chosen area is being shared by national and international participants. Now, I would like to say in this context that academicians receive as much as they give, as much as they share their views with others, and as much as they put in efforts in learning. I appeal to academic fraternity and galaxy of intellectuals, the best brains of India and abroad, that we should achieve excellence through innovation and quality education and research. As you all know, excellence depends upon 5 factors: self-motivated students, competent and committed faculty, pedagogical tools, infrastructure, and good governance. And we should also aim at excellence and everyday greatness. I always say these 2 important concepts, which are ultimates under the sun. Now, coming to everyday greatness is related to character and contribution. There are 8 time-tested and universally accepted principles which are included in everyday greatness. Since it is related to character and contribution, those 8 time-tested and universally accepted principles are vision, innovation, quality, humility, empathy, magnanimity, perseverance, and balance. Now, I always say that the academicians should become the center for power of academics and flavor of ethics, and the vigor and rigor of academic activities should add power and flavor to future generations. I also emphasize in this context that one should possess academic goal, research zeal, extension will, and governance skill. And ultimately, all academicians should serve the society through their scholarly activities. Now, coming to the seminar of biotechnology. The biotechnology can be considered as a tool for the improvement of human welfare, and its contribution to the welfare of the human beings is noteworthy and immense. And whether we talk of new invention of vaccine, or whether we talk of new crop varieties, or whether we discuss about improvements in livestock, poultry, meat, food, or improve the quality of milk, and even in war industry also, its contribution is immense. And biotechnology is playing a vital and very important role, and its significance cannot be underestimated, and it is taking commendable place in every field of science. Biotechnology has affected everyday life of human beings through its different inventions. And almost in every field of biotechnology, we have its products. Now, I congratulate for organizing this meaningful and purposeful international seminar, especially the convener, Professor Christopher Sir, Dean Research and Development, Organizing Secretary, Professor V. Rajendra Garu, Head Department of Biotechnology, and co-convener, Professor T. Murali Krishna Garu, Organizing Committee, Dr. V. Srilata, Mrs. G. Deepika, Mrs. D. Bhagyalakshmi, for their best efforts and making this seminar a grand success. I wholeheartedly invite the eminent resource persons to this seminar, Dr. Challa Suresh, Dr. R. Srinivas Rao, Dr. Shahnaz Majeed, Dr. Gulab Khan Rohela, for their expertise in their respective fields, and the topic chosen by them are very important and very relevant for the human welfare. Now, I conclude by saying that competition is not a curse for a creative academician. Championship is not a challenge for a committed academician. Uncertainty is neither an evil nor a devil for a dedicated academician. Flux is not a problem for a professional academician. World-class standards are maintained by worthy academicians. Winning is not at all a matter for an innovative academician. I wish the seminar a grand success. I thank one and all for providing me this opportunity. Thank you, one and all.
Thank you, sir, for your admirable and wonderful message given for us. Thank you so much, sir. Now, I would like to invite Dean Faculty of Science, Professor B.S. Anuradha Madam Garu, to address the participants.
Respected Vice Chancellor, Vice President of Chaitanya Deemed to be University, Dean Research, and the professors of all the departments, biotechnology, microbiology, and biotechnology and biochemistry, esteemed speakers of this function, and dear students and participants, a very good morning to you. I congratulate the team for organizing an international webinar with such eminent persons. And I'm very happy to also tell that both Srinivas and Gulab Khan are the alumni members of this university. So that is a real added feather to the cap. So both the students, both the speakers are our own students. So it is very nice to again have a get together of these people who are coming and giving a lecture on this particular platform. And it's a very great opportunity for all the students to listen to such eminent scholars, because it is, I think I can say to the students that one of the sincere form of respect is actually listening to what others have to say. So I request all the students to listen to, because you will not get an opportunity to listen to these great speakers every now and then. But we have made a platform for you to listen to such eminent speakers. And it is always said that better than 1,000 days of diligent study is one day with a very renowned scholar. So whatever we learn today, I'm sure it will help you in your future endeavors. I'm sure the deliberation will be very, very fruitful. We have the best speakers of the world coming together to tell to speak about their experiences, their research, which will go a long way to sow the seeds of research in our students. I congratulate once again to all the members of Biotechnology department for organizing this seminar. And I also thank you for giving me this opportunity to speak on this occasion. Thank you very much. Thank you very much, Rajendra sir.
Thank you, madam, for your wonderful, valuable words. Now I request Dr. Visrilekha ma'am to address the session for further event. Thank you, one and all.
Good morning, one and all. It's an honor and privilege for me to introduce today's keynote speaker, Dr. Challa Suresh sir. Dr. Suresh sir is a senior deputy director and scientist at the Department of Cell Biology Division at National Institute of Nutrition, which is a part of Indian Council of Medical Research, Hyderabad. He has an experience of more than 25 years at ICMR, and sir has obtained his master's degree in biochemistry from Sri Venkateshwara University, Tirupati, and received gold medal for being the first rank holder in the university. He completed his PhD in biochemical toxicology from the same university. He also obtained Doctor of Science in Biotechnology from Behrampur University, Orissa. He also obtained prestigious ICMR Young Biomedical Scientist Fellowship for the years 2014-2015. Currently, he is involved in major project funded by DBT, DST, and ICMR. He has research expertise in areas of nutritional biochemistry and biochemistry toxicology. He has more than 50 publications to his credit in national and international journals. 5 doctorates have been awarded under his guidance, and he is now supervising 5 research students. With this brief introduction, I would like to invite Dr. Suresh sir to deliver his talk on implications of lead on Alzheimer's progress, molecular approach. Over to you, sir.
Thank you very much. I'm grateful to Chaitanya College, where I've been associated since long, and as a board of studies member, and also I'm very much familiar with the students, also the faculty, especially Dr. Rajendra. And also I frequently visit that place for delivering several talks there. So I'm not new to this place actually. When Rajendra has asked me to deliver a talk, I'm more than ready to share my expertise. Shall I share my screen now?
Yes, sir.
Yes, sir.
Yes, sir, you can share.
Is my screen visible?
Yes, sir, it's visible, sir.
Yeah. So today, over a period of 20 years, I've been working in the area of lead toxicity and the close association of lead in development of the Alzheimer's. You all know that Alzheimer's is a neurodegenerative disorder, and people know that it causes a kind of dementia and memory loss, and it's very rampant in the age group of 40 to 60 years, and now the cases have been increasing in India also. Of course, people see that this is a very important problem at the Western world. No, it is not a problem at the Western world. And in India also, it is capturing the momentum. And in India also, the Alzheimer's cases have been increasing. And it's a very silent disease. Nobody knows about the progress of the disease. But when you see the reasons, still now, there is no particular reason being shown technically how the Alzheimer's progress can be detected, and how the Alzheimer's normally basically it comes into the picture. There are several factors. One of the factors is been working in the area of lead toxicity, and lead is said to be one of the important factors which can cause leads to the Alzheimer's progress, because in Alzheimer's ultimately the amyloid peptides gets deposited in the brain, and there will not be any signal transduction, and so that signal the one neuron to other neuron, the signal interruption will be there, so that the memory loss will be there. And I wonder how person living in a highly polluted environment where the lead is there in the environment, and lead, of course, lead is there everywhere in the environment through different kinds of sources, and how it can affect the Alzheimer's progress is one of the important key approaches, and the one of the prime areas at US also. And Indian government is also giving a very important approach to this addressing the issue of Alzheimer's progress. So therefore, for this particular, even though I have been given a short time, and my total whatever I did, and I try to make it as a gist, and I'll try to present here, which involves different kinds of techniques, which students also would like to know more of: flow cytometry, confocal, and fluorescence microscopy, and mark plotting, RT-PCR, several techniques, biotechnological approaches. Even though I'm a biochemist, I did my Doctor of Science in Biotechnology. And I think this presentation will help out. Basically, the target audience will be the students, and they will be more happy about this presentation. So my title goes like this: Implications of lead on Alzheimer's progress. There's a kind of approach where I published many papers. Let us see how it goes. And you all know that heavy metals are toxic and to human health, and they are non-degradable. Once they are into the environment and into the soil or whatever, wherever it is, they can't degrade, and they are very much toxic to the human system. And it shows serious health consequences. And basically, it's not only toxic, but it's also poisonous at even low concentrations. See, the most common heavy metals what we can see in the environment is lead, mercury, cadmium, and arsenic. And lead is said to be very toxic substance. So I have the government of India also giving importance, and our ICMR is also giving one of the test areas to have a light on the lead toxicity. So I have selected lead as one of the important tools. So pollutants by nature, consumption of contaminated drinking water, exposure to contaminated soils, and industrial waste. And these are all the sources for the kind of exposure of these heavy metals. And the food sources are such as vegetables, grains, fruits, fish, and shellfish. These can get contaminated by these heavy metals, and because of the surrounding water and soil gets contaminated through them, these can peep into the environment into the form of means into vegetables, grains, and whatever we eat. So therefore, it's a very important topic to address, because whatever we are consuming in a nutritional way, they have a kind of toxic substances. So therefore, they found to cause a kind of health effects, including reduced growth, development, cancer, and also several organs gets damaged. And most importantly, the nervous system is said to have a kind of important target for this kind of heavy metals. So, and ultimately, with even some extreme cases, we can see the death also. And these heavy metals, per se, lead, can be produced by the industrial activities and slowly gets deposited into the surrounding water and soil. And see, the arsenic, cadmium, chromium, mercury, lead, and nickel, these are said to be important heavy metals. And basically, they get into the body like through inhalation or ingestion. And also, this is one important route, and through absorption through the skin also. But I am not very much focused on other things. Now, let me look into the lead. Lead can be enter into the lead can enter into the body through inhalation and ingestion, and it can cause it's a neurotoxicant. It can cause neurotoxicity, and also it has some kind of effects on the lung and liver. And we don't know that our human beings are getting exposed to lead. Some kind of a loss of appetite and nausea also we can see. But most of the people, 90% of the people doesn't know that they have been implicated or inflicted with the lead exposure. So lead exposure can cause kind of loss of appetite also. And several organs, including kidney, liver, lungs can be damaged. But my area of interest is the nervous system. And I really show focus on the brain activity and how the brain functions with this lead exposure. So you know that environmental lead brain can get exposed to the environmental lead. It causes neurotoxicity, and it leads to the impairment of higher functions because brain is said to be one of the important target organ, and also very important organ in the body, which can play important role. And in infants and adolescents are get to be target groups with this exposure of lead. And continuous exposure of lead can lead to the growth and mental retardation, and also intellectual impairment and hyperactivity. In some cases, you can see even neurobehavioral changes also. And what are these kind of intellectual impairment and hyperactivity is kind of suppression of the cognition, learning, and memory functions, and also encephalopathy with coma and convulsions. These are the important aspects which are closely associated with the brain. So ultimately, the brain damage can be observed with the exposure of the lead. These are all invisible, and we don't know really. We are into the environment where mostly we are getting exposed to lead. So elevated levels of the blood, means lead in the blood, can diffuse through the blood-brain barrier, and it can enter into the brain, and it causes several cellular and neurological effects. And cellular effects are nothing but like intracellular mechanisms leading to the nerve cell death, and also the neurological effects is, like I said, encephalopathy, coma, convulsions, like brain damage, behavioral changes. These are all ultimately we can see in a long run. So as WHO says that there are 18 million people all over the world are affected with this kind of Alzheimer's problem, and there are 34 million it can reach to 34 million by 2025. And in India, the people getting exposed are 4 million by 2025. So this can cause, as I said already, several studies in the rodent models and also rats in mice saying that it has got early exposure of the lead to the brain causes irreversible damage. So therefore, lead is considered to be an important possible risk factor for the pathology of Alzheimer's disease. So, you know, the Alzheimer's disease is nothing but this progressive neurodegenerative disorder where you can see the loss of neurons and the abundance of intraneurofibril tangles associated with the extracellular deposition of the beta amyloid peptide. And the classical case is the formation of the plaques. And the loss of cognitive functions, dementia, and death, the ultimate effects of the Alzheimer's disease. So where in Alzheimer's, you can see the tangles, neurofibrillary tangles formation, which aggregates into the brain, and which is closely associated with the tau protein, and also the amyloid precursor protein, which is formed there, with sequential lysis of the amyloid precursor protein through the beta secretase and gamma secretase leads to the aggregation of the amyloid peptides. So the amyloid peptides gets deposited in the brain, and it causes a kind of intervention in the signal transduction pathways, and it causes inflammation, and also cell cycle also gets affected. These are all the parameters which we can closely focus now, and what could be the causes of the lead in making the Alzheimer's progression. So the mechanism by which the plaques and tangles, which is the classical situation in the Alzheimer's, is still not known. We don't know anything, but several people are working in the area, but they still now, nobody has given a proper mechanism why these plaques are getting deposited and these tangles are formed in the brain. And one of the classical situation is the formation of the beta amyloid peptide, which ultimately gets deposited because of the lysis of the amyloid precursor protein, that is APP, into the brain, and which ultimately causes the irreversible damage like neurotoxicity and oxidative damage. And as I said earlier, and it causes inflammation also. These are all the things which we can address. And in my later part of my presentation, I'll try to look into the different kinds of neuroprotective compounds which by which you can get protected, our brain can be protected. And the toxicity being resulted by the combination of the lead and amyloid peptides. No studies were there. I'm the first person of its kind working on this area for the last 20 years. And the lead is said to be one of the risk factors for the amyloid, means amyloid peptide deposition, ultimately the Alzheimer's. We see here the Alzheimer's disease is a classical situation where the neuronal plaques will be formed here. Here, this one, and neurofibrillary tangles also can see, and the plaques and neurofibrillary tangles under a confocal microscope appear so like this. And it's a classical case where the loss of memory and the cognitive impairment, neurobehavioral changes can be observed. This all reflects, this picture reflects all kinds of things. See, the size of the brain gets reduced into very small, so that we can't keep the things in the brain, memory loss will be there. I request all the people get mute, no? Some sound is coming. So the
Sir, మీరు unmute చేసుకోండి sir.
Yeah, I'm audible now.
Yes sir. Yes sir.
Okay. So the amyloid peptides induce toxicity, which causes the neuronal death, and which involves complicated pathways here, including mitochondria and neuronal cells and the synaptic, where the synapsis happens here, and also the altered calcium signaling pathway, which ultimately leads to the inflammation. These are all the things which I'm going to address now in my study. See, our previous reports, and based on our previous reports during 2005, 8, and 12, which we did, our lab did and our group did, and where we found that lead causes the neuronal cell death. Based on those reports, we further studied to see the effects of lead in the Alzheimer's situation. So therefore, we made a hypothesis saying that for every study, we need to have a hypothesis before taking up our study. And our hypothesis is the synergistic neurotoxic effects of lead and amyloid peptides can be mitigated or reduced using naturally available neuroprotective compounds. So not only how the toxicity is being done, the investigating that component, even we try to have a kind of address by using different kinds of bioactive compounds that are said to be antioxidants. The battery of compounds again is lead induced toxicity in the Alzheimer's situation. So So the primary objectives of my study is I'm audible?
Yes sir. Yes sir.
Okay. So to study the synergistic effects of lead and amyloid peptides in human brain cells and their effects on different biomarkers like inflammation, signal transduction, oxidative stress, and also the apoptosis. The apoptosis is said to be one of the important the target mechanism where ultimately the cell gets into the death situation, which is an irreversible damage. That ultimately death means there is nothing we can get back. So apoptosis is one of the reasons, the induced apoptosis, that also we try to address here, and to identify several intracellular mechanisms of naturally available neuroprotective compounds and their effects against the toxicity being induced by the lead and also amyloid peptides. So we the plan of execution is how we have executed our plan. Whatever the data I'm presenting is the gist of the work which I have been carrying out. Briefly, I'm presenting here. And we have taken SHSY cells, brain cells, and we try to grow them in the laboratory because it's a model for the brain. And also we try to see the exposure of lead and to these in different concentration to these brain cells, and also the amyloid peptides like 140 and 2535. These two are said to be important amyloid peptides, which you can see the brain. So those amyloid peptides also we got tried to expose to the brain cells, and we did time-dependent, dose-dependent studies with these two, and also we tried to find out the IC50 values and the cell morphology also we observed through using like a phase contrast and scanning electron microscope. And these are all the battery of techniques where we have used advanced techniques like flow cytometry, confocal scanning, and RT-PCR, and several techniques we have used because our lab is very much having the facility. So these are all the methods which we followed, this methodologies that's not required here. And these are all the compounds. We have a battery of compounds. We have tried to see the screen these compounds like epigallocatechin, gallate, genesin, quercetin, resveratrol, curcumin, fraxetin, epigenin, magnolol, and pinosumbrine, coidine, garlic extract. Among ways, the epigallocatechin gallate is said to have one of the important the molecule which we consume every day in green tea, and the leaves, the important sources is the apple and also the leaves of the bihi. So which is said to have an antioxidant nature, and which can inhibit the ROS level, that is oxidative stress, and it can inhibit the apoptotic progression, and also try to address the amyloid beta fibril formation and oligomerization. It can interrupt in the formation of the oligomerization, and it's, of course, it has because it's said to be an antioxidant, potential antioxidant. It has got anti-inflammatory effects also, and these are all the structures of these compounds, and they're all said to be the polyphenols basically. And you see the all polyphenols in nature, whatever available polyphenols, they are said to be antioxidants, and they have several effects in the body. And EGCG is extracted from the green tea, the fraxetin from the fruits, and the magnolol from the mango, and the quinidine is from the curry leaves, and they are said to have a protection against the amyloid peptides. And also they are antioxidant in nature and anti-tumor, anti-inflammatory, and anti-diabetic, and they have the effects in enhancing the neuronal proliferation and function. So of course here even though we have data on fraxetin, coidine, magnolol, I'm going to present only with the data obtained through the EGCG because time is not sufficient here. So the gist of the effects of the EGCG against the Alzheimer's, amyloid peptide induced toxicity in the brain cells, I'll try to project here. So the findings we have studied initially, we have exposed lead to the brain from 1000 micromoles to 0.0001 micromoles, and this is a dose dependent or concentration dependent study, and we found that this is having kind of effects in reducing the cell viability. That means cell death has been happening at micromole level between 10 and 1 micromoles, and then we have narrowed down and try to see the effects of this lead on the brain cells at between 10 and 1 micromole levels. And because this one is considered to be important study here in fixing the dose before conducting all kinds of experiments, because whatever the experiments further we are going to conduct, it has to be to begin with good data and to fix the dose. So the 5 micromoles is said to be the IC50, which we have fixed for further studies. And also we have studied not only the concentration dependent, but the time dependent also, the 12 hours, 24 hours, and 48 hours. And we found that the 50% of decrease at, of course, 5 micromoles, but the effective concentration is at the 24 hours. So we have taken the 5 micromoles as IC50, and also 24 hours is the time period for further studies. And also because amyloid peptides, we can't take the brain and we can just study because it's a live human brain, we can't take. So therefore, we have taken the synthetic amyloid peptides like AP, amyloid peptide 2535, which is available from the Sigma, and also amyloid peptide 140. These two are the important peptides which gets deposited in the brain during the Alzheimer's progression. So therefore, we want to mimic the situation in the brain cells. So we have taken AP over 25, 35, and 140, and we try to fix the doses here also. Like we fix because these two are said to be toxicants to the brain cells. So we try to have a kind of concentration dependent study, dose dependent. And also here, the 8 micromoles is said to be the IC50 for the 25-35, and also 60 micromoles are said to be the IC50 for the 140. So these two, 8 micromoles of 25-35 and 60 micromoles of the 140 as the model for the Alzheimer's situation we have selected. We try to use these concentration for further studies. And here also we have seen the time dependent study like in lead exposure, where we have found that 24 hours is said to have a kind of effect, and 50% death where IC50 observed for 25-35 is said to be 8 micromoles, and 140 is said to be 60 micromoles. So this is what we can observe under the microscope, like phase contrast microscope. There we can see the control group and the lead exposed, and amyloid peptide 25-35 exposed, and AP 140, and the combinations lead 140 lead 25-35 lead with 140 25-35 and 140 25-35. These are the combinations where the effective concentrations, how it is going to affect the brain cells we have studied. And here we found that clearly the control group is having connections between the cells, and the loss of connections in other cells, and the cells become the conical cells become circular shape, and we can see the decrease in cell number and the loss of neuritis, and the cell texture and shape also been changed morphologically, and ultimately the cell lysis is happening, because once cell lysis is there, that means ultimately the apoptosis comes into picture. That means cell is undergoing death. See, this one is Biogen scanning electron microscopy. We can see here the control groups are like other groups, and the increased bubbling which indicates the apoptosis, and increased membrane protrusions here, which also says that it is undergoing a kind of lysis, and the disconnection between the cells and the loss of cell texture. Here we can see the loss of cell texture, and also the shape, which regular shape is been changed to a kind of like balloon-like structure, and ultimately we can see the cell lysis, which has been authenticated from the data of the phase contrast microscopy. And even we have seen whether the cells can take up the DNA. Whether the DNA dye, this propidium iodide is one of the dye, which is a marker to see the dead cells. So whether when you expose this propidium iodide, how the cells take up, because if cells takes up, that means the indicator for the dead cells. If more of toxicity is there, the cell dye will take up into the cells, so that we can see the death, more of death. So we can see in the control group there is no death, and in the toxic death we observed in the lead and 140, 25-35, the combinations, all kinds of exposure where more of dye showing that the cell death is there. And see here how the amyloid peptides get secreted. The simple enzyme is the beta secretase, that is the base 1 enzyme, and the base 1 enzyme also we have tested by exposure to the lead and 140, 25-35 combinations. And because the major triggering element in the progression of the Alzheimer's disease is the formation of the amyloid peptides, where if you have more of amyloid peptides get deposited in the brain, that means the beta secretase levels are also more. That means we have to check the beta secretase level before going into the amyloid peptide deposition. So if the more levels of the base one, that is beta secretase levels, if you observe, that means more of the amyloid peptide. This the Western blotting, the method, what we observed here, the bands shows that the more of formation of the beta secretase, that means more of we can anticipate. It's indirectly saying that the more of amyloid peptides getting deposited here. So this this indicates that the beta secretase is playing an important role in getting deposition with the lead also. Of course, in amyloid peptide induced toxicity that is been there, beta secretase levels will go up. But the fascinating thing is lead is also causing one of the important toxic effects on the secretase, because secretase levels is also going up with the lead exposure. So here the antioxidant markers also we have tested whether the cells are undergoing oxidative naturally. When the inflammation, oxidative stress is there, that means you can see the more of inflammation, and this is closely associated. More of oxidative stress leads to the more of inflammation and the apoptosis also. So therefore, we have tested with the oxidative stress markers like ROS, reactive oxygen species, nitric oxide, and also the MDA levels, where we can see the alterations with the exposure of the lead and amyloid peptides, which indicates that the cells are undergoing a severe oxidative stress. That means body has to have more of free radical generation. That means free radical damage is there for the membranes. So here, apart from the other markers, the important marker is the total glutathione and also the catalase, SOD, superoxide dismutase, and vitamin C. Vitamin C, most of the people doesn't know that vitamin C also plays an important role as an antioxidant. So in recent situation where COVID situation, vitamin C 1,000 mg per day has been prescribed because of its nature as an antioxidant. Here, when you are studying oxidative stress parameters, vitamin C also one of the important marker. Of course, vitamin E also tocopherol, alpha tocopherol is also one of the important oxidative antioxidant. That data I'm not projecting here. That also gets altered here. And see whether apoptotic proteins like BAX and BCL2. BAX is a pro-apoptotic protein marker and BCL2 is an anti-apoptotic marker. How this go hand in glove during the situations where toxicity is there. See, we have tested with both BAX and BCL2. They are adversely being affected here. And the levels, alterations in the levels of the BAX and BCL2 indirectly indicates that the caspase 3 levels have been altered. You know, caspase 3 is one of the enzyme, important enzyme in the caspase pathway. It is the ultimate execution of the apoptotic pathway. If you measure the caspase 3 levels, it indicates that the elevated levels of caspase 3 indicates that it is the cells are undergoing death through the apoptosis. So these levels of BAX and BCL2 automatically the levels alterations of these two levels triggers the elevated levels of caspase 3. So that is being observed here with the exposure of lead and amyloid peptides. And here the BAX BCL2 ratio is one of the important ratio where anybody in the world we can just test with the BAX BCL2 ratio, saying that how much apoptosis has been happening in the system. So the BAX BCL2 ratio, increase in the ratio indicates that the cells are undergoing apoptosis, means premature apoptosis. That means the induction of the apoptosis. That means cells are undergoing death. So we have used in advance to ascertain that whether the BCL2 or whatever it may be is showing showing effects on the apoptosis. We have used advanced technique like flow cytometry to test upon whether the cells are undergoing death because of the apoptosis. Yes, really this slide indicates that the flow cytometric images indicates that the combination of the lead and amyloid peptide 140 and 125 causes the damage around 24.9%. Of course, the necrosis is also one of the process. You all know about the necrosis. If you don't know, just go through about necrosis. But necrosis is not similar kind of that apoptosis. But surprisingly here, the combination of the necrosis and apoptosis has been observed here with the combination of the toxicity. And as I said earlier, caspase 3, one of the execution marker, and which plays an important role in the intrinsic pathway of the apoptosis. is also being tested out, then increased levels of the caspase 3 indicates that the cell is severely undergoing death. So it's irreversible damage. So the apoptosis is being controlled by the caspase pathway, caspase 3, caspase 9, 12, 8, all these markers, but caspase 3 is an executioner. So if you can study and the results of the caspase 3 indicates that the cells are undergoing death. And we have even tested the cell cycle. The cell cycle is one of the important tool in these cells, where these phases like G1S and G2 phase are there. And if S phase is being altered, then that means the cell are arresting the cells in the S phase. So the lead and amylide peptides are showing a kind of arresting of S phase. So that's lead is showing 40 to 40.8%, and the combination of the lead and amylide peptide shows that the 68.9%. That means the cell synthetic phase has been getting arrested, so that more of cell death is there. That indicates that cell death is there, and more number of cells are not getting multiplied. So, and it is, as I said earlier, the apoptosis, oxidative stress and inflammation are closely associated with the mitochondria also. So one of the important mitochondrial protein is voltage dependent anion channel protein, that is VDAC1, which can be a marker to study the mitochondrial mediated apoptosis, and the overexpression of the VDAC leads to the cytochrome C release. You all know about cytochrome C. Cytochrome C plays an important target group for the release of these proteins from the cell. That means ultimately the cell is undergoing cell lysis. So that means the VDAC is being triggered, triggering protein here, and which mediates the release of the cytochrome C and leads to the apoptosis. So we have studied the VDAC protein also. And here the expression, the mitochondrial protein expression like VDAC and the cytochrome C has been the overexpression being observed here with the exposure of the lead and amylide peptides, showing that the apoptosis is mitochondria mediated. And see, as I told already, VDAC transports ions, calcium, cholesterol, and metabolites across the outer mitochondrial membrane and participates in the release of mitochondrial pro-apoptic protein. Any kinds of alterations in the VDAC1 leads to the release of the more of cytochrome C, which ultimately leads to the apoptosis. And the other kind of things is calcium. Calcium dysregulation of the calcium levels in the body is an important marker for the plasticity. How the cells getting undergoing whatever the signaling pathway associated with the calmodulin and calcium is very close. So alterations at the alterations in the cell, that means the calcium dysregulation is also playing an important role. That is synaptophysin is an important marker that also been studied, and the GAP43, that is growth associated protein, also been altered with the help of the PKC, protein kinase C levels. And here, how the calcium is getting affected. See, lead can replace intracellular calcium, and the altered neuronal calcium homeostasis plays an important role in the neuronal degeneration. So the intracellular calcium levels have been, we have used flow cytometry to see the intracellular calcium levels by using a flow 3 acetoxymethyl ester dye. If you put that dye, automatically it gets bind to the calcium, and we can see the levels of the calcium. And the decreased levels of the calcium levels have been observed in the cells. That means it is causing a dysregulation of the calcium, and ultimately the calmodulin levels also. So the inflammatory markers, I was been talking about the inflammation. Inflammation is involved in the neuronal death. See, with the different kinds of pro-inflammatory factors like TNF alpha and interleukin 1 beta and interleukin 6. These are said to be very much toxic to the neurons. And when this kind of exposure of the amylide peptides and the brain led to the brain cells, causing the overexpression of these kind of proteins like like inflammatory markers. I'm not saying that protein is a marker. And TNF alpha, IL-1B, and IL-6, they are being upregulated in the body. That's showing that it is also causing inflammation. And the prostaglandin E2 and isoprostane 8 isoprostane, These two are very important associated with the fatty acid metabolism, and these are the important markers for the overexpression of means the inflammation is causing of the increased levels of the inflammation. So the prostaglandin E2 levels, if they get increased, the body says that the inflammation is there, and automatically the isoprostaglandin levels also gets altered here, because the prostaglandins are fatty acid derivatives of arachidonic acid. You know about arachidonic acid, and which are closely associated with the membranes of the phospholipids, and also COX enzyme activity and that pathway. And also isoprostaglandins are the family of icosanoids, and also is a non-enzymatic origin produced by the random oxidation of the membrane phospholipids and by oxygen free radicals. More of oxygen free radicals and more of inflammation, and the levels of the isoprostaglandin level is also being overexpressed. And also the MAP kinase levels, MAP kinase and P53, and it also the COX2 expression is also been increased here, and also the MAP kinase levels have been also been altered, so that the inflammation results with the AB toxicity is closely associated with the lead. So lead can, of course, lead can enhance the toxicity of amyloid peptides. So that means the Alzheimer's progress is adversely affected by the lead exposure. So then with this data, first this is the gist of the data. I have a lot of data in that area, and how production can be done. Ultimately, when you are doing some work, there should be a translational aspect for the research. So therefore, we have tested with the different kinds of battery of the mnudoproductive compounds. Among those, the EGCG is one of the important compound, like epigallocatechin gallate. And as I said earlier, we have tested with the EGCG and dose-dependent studies. We found that 50 micromoles is suitable for the further studies, and also we try to see whether EGCG treated cells can protect the cell amyloid and lead to exposure toxicity. Yes, definitely the cell viability has been increased with the exposure of the EGCG. You can see here the EGCG group, this is untreated group. The viability of the cells have been increased. This can simply we can test with the MTT assay, and also we tested with the apoptosis by using flow cytometry, where the apoptotic proteins levels have been reduced here, and we say that the EGCG is causing kind of neuroprotection. And here you can see the apoptosis with EGCG has been with the exposure of the EGCG, the levels have been decreased very closely, showing that it can arrest the apoptosis and it can intervene and closely can reduce the apoptotic cell death. And also the oxidative stress, like ROS we have tested here, and the reactive oxygen species. The levels have been reactive oxygen species, that means free radical damage, how much extent is been there. We test with the lead and amyloid peptides. Here EGCG try to control that. The more of free radical levels have been reduced here, showing that it can effectively access antioxidant against the lead-induced toxicity. And also the MDA level, similar kind of effects we can see here. And the EGCG exposed cells shows that reduced malmaldehyde levels, showing that it can reduce the oxidative stress. And also here, the prostaglandin, I told you earlier, the prostaglandin plays an important role in the inflammation, and the reduction in the PGE2 says that it is reversing the situation of the what you call that inflammation. And also the cell cycle, where the S phase, I was talking of the synthetic phase, which can the arrest of the cells at the synthetic phase has been reversed here. And the EGCG treatment shows that it kind of protection and the synthetic phase arrest is been drastically reduced here, that we can see here. And the thioflavin T fluorescence, that is a kind of thioflavin T is a fluorescent dye which can bind to the amyloid proteins, where the EGCG levels, the EGCG when exposed the epigallocatechin level, the thiofluorescent fluorescence levels have been, the intensity of the fluorescence has been reduced. And also the Congo red, one of the important marker for the fibrillar and non-fibrillar materials, and is a direct dye which can the benzidine based anionic benzo dye with the intensity of the Congo red also get decreased, showing that the more the deposition of the amyloid peptide has been reduced drastically with the exposure of the EGCG. And you see the fluorescent microscopy here by using DAPI. DAPI anti-APP means anti-amyloid precursor protein, and merge, which says that the amyloid peptide deposition is being reduced with the help of EGCG. So that means the EGCG plays as a protective agent, neuroprotective agent. And also, like previously I said, the BAX and BCL2, cytochrome C, these all we tested whether this can protect the cells from the being damage being happened with the apoptosis. Yes, the EGCG is trying to protect the brain cells. And also, similarly, we have seen the calcium levels. Calcium levels have been altered, and the reduced calcium levels have been improved. And also the CAM kinase levels, PKG, protein kinase levels, where this exposure of EGCG leads to the improved levels of EGCG, this CAM kinase and protein kinase, and which is closely associated with the calmodulin, the expression. And also the GAP43, as I told you, growth associated protein, that is 43, and also synaptophysin. These two are important markers for the synaptic plasticity, where the signal transduction is been happening properly or not. These two markers have been adversely reversed with the exposure of the EGCG, and showing that more of signaling is happening. That means memory loss can be reduced. And the ultimately what I can say is the latent Alzheimer's situation can alter the cell viability, cytotoxicity by lactate dehydrogenase, and we can reduce the neurite length also, and the lipid peroxidation, neuronal markers like B3 tubulin, GAP43 gets altered, and the intracellular signaling of the abnormal processing of the APP, that is amyloid precursor protein, and the dysregulation of the calcium homeostasis here, and oxidative stress. Here, the abnormal processing of the APP is increased levels of base enzymes is reversed here, and increased deposition of the beta amyloid peptide, thioplovin, and the beta amyloid peptides. The top protein levels have all been observed through confocal, and saying that these can be altered with the exposure of the EGCG. And the intracellular calcium levels, the PKG activation, and decreased levels of calmodulin, VDAC, decreased GAP43 and beta 3, tubulin and synaptophysin levels have also been altered. And the oxidative stress, when you see the oxidative stress markers like RYS, NYS, GSH catalase, all this been altered, and the caspase 3 cytochrome C levels, which is closely associated with the mitochondria, has also been altered, and the decreased NF kappa B also been observed, which ultimately leads to the neuronal degeneration and cell death. And with the exposure of the EGCG, we observed that everything is been altered, reversed back, and saying that it can easily protect the neuronal cells. That means it is having a kind of a protective approach, and even we can prescribe that one. Preclinical testing also has been done, and it has got the effects of the inflammation at the membrane level, and also the mitochondrial level, and also signaling where it is neuronal signal is happening, and also that synaptic levels. So which all been reversed with the exposure of the EGCG, and ultimately we can prevent the neuronal death. And these are all the publications what we have with this, with all kinds of impact factors like 5, 4.55, more than 5 impact factor journals, like chemical biological interactions, neuronutritional neurosciences, all these kind of journals were published. And these are all the publications we had last 5 years. Thank you very much.
Thank you, sir, for giving your valuable insights on the topic. Thank you very much for the wonderful session on lead toxicity and detoxification as well. And so there are many compliments in the chat box for you, sir. And we have questions, few questions.
Yeah. You can ask the questions.
I would request participants to unmute yourself, and if you have any queries, you can just ask sir. All the participants are requested. If you have any queries, you please unmute yourself and you can clarify your doubts.
If you have any doubts, you can contact me by mail also. It's no need. Thank you very much for giving me a wonderful opportunity. If you don't have any further questions.
Thank you, sir. Thank you so much for your excellent presentation on Alzheimer. It was really a very interesting lecture today, sir, after your data. Thank you so much, sir. Thank you.
Thank you.
Thank you, sir. Thank you once again. So now I request Dr. B. Srilata Madam, Head Department of Microbiology, to introduce our next speaker. Dr. Srinivas, sir.
Good morning, everyone. I feel extremely honored to introduce today's next speaker, Pride alumni, my friend, Dr. Srinivas Rao Ravilla, a renowned microbiologist and senior fermentation scientist at Institute of Biological, Environmental and Rural Sciences, Aberystwyth University, UK. He is our Pride alumni, graduated from our Chaitanya University in 1995, obtained his master's from University of Madras, completed his PhD from Osmania University, Hyderabad, in collaboration with IICT and Gayatri Group of Industries, and postdoc from CCMB, Hyderabad. Dr. Srinivas has more than 21 years of research experience. He actively involved as PI and co-investigator on 15 of UK, European Union and international grants such as Innovate UK, Climate KIC, Bacon, BBSRC, European Biogas, UK India Newton Baba Industrial Waste Challenge grants. Most of the projects in collaboration with industries, and their total value exceeds over 20 million pounds. Srinivas also published more than 60 peer-reviewed international publications with high impact factor with a H index of 31. And he received so many awards like the European Union Commission's 2014 Regio Stars Award for Sustainable Growth, EU Bacon Project, European Bio-Based Chemical Partnership of the year 2016 regarding Climate KIC Project, Outstanding Pay Progression Award by Rothamsted Research in 2009 UK, Global Initial Academic Network Faculty, Ministry of Human Resource Development, Government of India, New Delhi. He is also a scientific project
title page title page పెట్టాలి కదా.
Hello.
Ma'am, you went on mute, ma'am.
No, madam, you are not audible. Unmute yourself, madam.
It's okay. I think
You are not audible, madam. Srilata, madam, please unmute.
You're in Rajendra.
I now request Dr. Srinivas sir to deliver his talk on.
Okay. Can you hear me?
Yes, sir. We can hear you, sir.
Okay. I would like to share my screen and then.
Yeah.
I think it's disabled, I think, screen sharing. It's showing host disabled participant screen sharing, sorry.
Yeah, Rajendra sir, kindly allow sharing by Dr. Srinivas sir. Screen sharing, please.
I am unable to share the screen. It's showing disabled. Sorry.
Yeah.
Okay, sir. I will make a pause, sir. Please. So now you can share, sir. Srinivas sir.
Okay. Can you see now? Can you see him now?
Sir, yes, sir. It is visible, sir.
Okay. Good morning, and thank you very much for inviting and giving me an opportunity to present today regarding my talk on role of microbes in human welfare. Thank you very much, sir. Thank you very much for organizing committee and everyone. And it's an honor to be presenting here with you all. So I work at IBUS, Aberystwyth University. So this is the institute. It is like our CSR institute, like CCMBR, ISCT, but we are part of the Aberystwyth University. It's an independent department. It's more than 100 years old, where the scientists work more on grass breeding and as well as oats. So most of the farmers in UK and Europe, they use the seeds from the institutes. So recently celebrated around 100 years of experience of the institute. So we now the next level, so we doing much of the biorefining, where they develop the grass varieties they use to get more milk and meat from cows, but the same varieties can be used to produce ethanol, other value-added products like proteins from the grass varieties. That's what it started, more refining of the grass to value-added products. Before I getting there, I would like to start, as I said, that this sort of the experience started from Hyderabad, and now we are at Aberystwyth. So I would like to give you some sort of information on how I started my work on yeast strains or developing yeast-based technologies. So you all might be knowing the Shanta Biotechnics. They started in 1992 when it's like a university industry partnership, like a small partnership in 1993. So they came to the Osmania University, and later they thought that it's better to go to Center for Cellular and Molecular Biology to develop the technology. So it started in 1992, and CCMB initiated work on developing vaccine, to develop vaccines. And they got an investment from Oman initially, and they started in 1995. So that's what it started. CCMB given the technology to produce the vaccines using bacterial strains at the time. First DNA recombinant DNA technology to Shanta. So that's what it started in 1995. And then in 2005, after 10 years, they're selling like millions of doses of different vaccines. So then they thought, instead of using bacterial strains, it's good to use some sort of the yeast strains to produce vaccines. That's what they thought. So they started in 2005. We started with Shanta working on the yeast expression system, part of the NIMITLI program, to develop some yeast expression systems for the vaccine production. While we are under development, I think there is a big transition from 2005 to 2007, where the multinational companies, they've taken over the Shanta. At the time, the value is around 784 million, like around 4,000 crores. So in a span of time, it started from $1 million and it went to $784 million in a few years of time. That's the power of the technology if you use microbes in a proper way. As we see now at the time of pandemic, how the vaccines being produced so rapidly and rolled out all over the world. So the technology is so important. Those are microbial based, maybe bacterial or yeast expression systems, but that's what the use of the yeast and bacteria for value-added products. So in terms of the yeast, because I've been working on yeast for 20 years, so I would like to give you some sort of the ideas how we can use for the yeast for different variety of products. It's a huge market. You can see by 2025 with a 10% growth rate, it's around 45,000 crores. It might be in the baking industry, it might be in the ethanol, not only producing the beer, but non-alcoholic beverages and the ethanol for blending into the petroleum, and also for pharmaceutical products where you can use yeast extract. So there is a lot of versatile use of microorganisms in industry, especially from the yeast. So that's what it is. It's a huge market globally, and a lot of work going on internationally in terms of basic research and as well as advanced research on the yeast. So to start with, as I said, we started this program, part of the NIMITLI program from CSR funding, which collaboration with Shanta and CCMB, to start a novel methyl expression system. So initially, that's the idea. So to isolate a methanol utilizing yeast strain, methylotropic yeast strain means it should utilize methanol as a carbon source. Then there is a genes, alcohol oxidase genes that can drive any sort of the genes to produce vaccines. So to very strong known promoters to drive the genes. So we have to isolate a new methanol utilizing strain to develop the yeast expression system. So that is the challenge at the time. So we started isolating the strains using liquid and solid media exclusively to isolate methanol utilizing strains using as a methanol carbon source with yeast nitrogen base. So that's what the simple start. So it's easy to isolate some yeast, but it's very difficult to isolate a particular yeast if you are interested in to very niche market. So we started screening yeast from all over the samples from all over the places. At the time, I think in a span of 1.5 years, we have isolated more than 2,000 yeast strains. So once you isolate the yeast, you can isolate from soil, you can isolate from diversified environment. I'm going to speak about that. But once you isolate the strains, then you need to identify what sort of the species it is. So there are known around 1,500 known yeast species so far. So you can identify the thing with a molecular biological technique. So you can sequence the ITS region of the yeast, the DNA from the yeast, and you will be identified what sort of the yeast species it is. Because when you go back and see on the colony morphology, it's very difficult to say what sort of the species it is. You might have a yeast, and you can confirm that by looking into the microbiological examinations. So you can purify the yeast from the plates, and then the next level going to be identification of the yeast. So that is very, very crucial to know the little bit background of the yeast strain. So you can use some simple kits if you have. If not, you have to go for this molecular characterization. So you're going to isolate the DNA from the bacteria or yeast, and then you're going to amplify the rDNA genes to know what species it is. So you're going to use some primers to identify that particular gene region. So we can use exclusively for fungi. It's either NL1 or NL4 or ITS specific targeted PCR primers to amplify the particular region of the DNA. So we can use a different set of primers to amplify the particular region of the DNA. So once you get that, then you're gonna sequence it to know the further analysis. So initially you can use a very small fragment, like 100 to 1000 base pairs. So that sort of the region we can identify. Even for yeast, 600 base pairs would be fine. But if you use ITS primers, you're going to get more than 1000 base pairs, so 1 kb. So at the time, that is really advanced. But now we have a lot of advances in sequencing technologies where we can even sequence the whole human genome in few hours time. So that's what the technology is developed. I was talking about 2000, 2001, but now we are in 2021. So in 20 years, there is a lot of improvement in terms of the sequencing technologies, where we call it as the next generation sequencing technologies and genotypes. So that's more useful, these rapid technologies, to identify the strains from all over the world. So once you get that fragment of 500 to 600 base pairs, then you can blast into the database, and then you might be knowing what species it is. For example, once we amplify the DNA, so here you can see some 500 to 600 base pairs, then you're going to blast into the gene bank. Then it will retrieve what sort of the species is. For example, a Candida species Y28, that's what the designation we give different strains. For example, Candida YS12A. So this shows that it's close to Candida species. And if you see the bottom one, it's going to be Escherichia species. So that's what the information you get. Once you isolate the DNA, then isolate the specific region, and then you blast it to the gene bank. Then gene bank will retrieve some sort of the information about the background information of the strain, how close it is to previous existing species, or it might be a new species. So that's what the information you're going to get out of it. And that's also show you what sort of the close relevant to the previous organisms. For example, in this, we have the strains YS9, YS6, 44, 43, 54, and that will tell that how close it is the previous known organisms. Like, for example, YS54, it's very close to Pichia carabica or Pichia galamondi. So that means it's a Pichia species. That's what you get to know if you do this sort of a little bit of bioinformatics analysis based on the sequences. So it's very, very nice. It's also a little bit tedious, because if it is only 1 or 2 strains, you can characterize. But if you are handling around hundreds of thousands of strains, it's very, very tedious. So every day we used to isolate around 20-25 strains and sequence it within a week, get the DNA, get the PCR done, and then send it for sequencing, get the information, blast into the database, and then see what it is. So if it is a known species, then you have to accept it as a known reported species. But as I said, we have around 15,000 yeast species so far reported. So when we're screening this 2,000 yeast strains for the methanol utilization, so we got some novel species. So we named after Hyderabad, that is the first strain. So we call it as a Candida hyderbadensis. So that's isolated from the grapes from the market. So that's what we reported, the first new yeast species from Hyderabad. It's called Candida hyderbadensis. And another one we reported from papaya fruit, that's for Sisymensis, is honoring the CCMP where I was working at the time. But it's not only limited to that. We have gone to very weird sort of samples and then to isolate the yeast. So for example, here, one day, fine morning, my boss bought a small snake. It's a trinket snake. So he said that isolate some yeast from that. So because the snakes eats everything and they digest everything. So in the gut, there might be some weird microorganisms. So we thought of doing that. So we have reported Blastobotrytis serpentis. Because it's from the snake. So there is also some bacteria at the time we have isolated from deep sea. So that is also a new Microbacterium indicum. So you get a novel species. That means those are very novel. They might produce novel products or the enzymes, or they might have some sort of the use if we screen further, if we understand further about that particular species. So as I said, we gone very extreme environments to get the samples. As I said, we got the samples from the deep sea, and then you can see here, even try to isolate at the time from the high altitude. It's like sending some sort of the payloads to 20, 30 kilometer high in the space and get the samples. So that's we done with the ISRO. At the time, they sent a balloon to that high altitude, and there is a cryogenic probes, automatic probes to get the air at that altitude and then get back the payload and chase with the helicopter. It's like a James Bond movie. Anyway, I haven't been there, but that's what it is. It's very expensive to collect the samples sometimes at that altitude, but you get a very, very novel spores forming organisms, very high radiation tolerant organisms. So that's what 20 to 40 kilometers high altitude. So there is a samples when I joined in the week 1. So we got the cryogenic vials and we try to isolate some bacteria from that. And later they reported quite a lot of new genus and species from that high altitude. And also we got some samples from the frozen lake of Himalayas. Also the lab people used to go to Antarctica to get some samples from Antarctica. So these are all very, very extreme environments to see what sort of the novel microorganisms. For example, if you go to the cold region like Himalayas, where the frozen lake, you have the yeast that can grow at 4 degrees or 5 degrees centigrade, and they produce some sort of the cold enzymes that might be used for biorefineries. So it's not just some soil samples. So if you really want to isolate some very exclusive microorganisms, then you have to look for very diverse environment. And the basic skills, whatever the sample it's going to be, once it comes to the lab, then it's your skill how you're going to purify the yeast, or how you're going to isolate the bacteria, and how you're going to purify them. So those are all basic microbiological techniques that is very, very crucial to do any sort of the work. So at the time of graduation at college, we do have a very good teachers like Anuradha Madam and as well as Dr. Rajkumari. They taught a lot at the time to know the basic microbiology. It's very, very new institution at the time, and we are very new to that. But it's a fascinating science to learn. First time microbiology started in that region, especially in Telangana, especially in Hanamkonda. We are the first batch people. So everyone will be fascinated. It's quite novel, new. So the learning process from there, it's went on the next 20 years, layer by layer by layer. So you can't just get mastered in one go. But we learned a lot of basic, even from gram staining to any sort of work, a little bit of fermentation. So that's what it started, very, very humble start. And then it's went on a lot later stage. It's just enthusiasm, how you can do the science. So that's continued. And once I left the Hyderabad in 2007, when I moved to UK, even though my project's not exclusively on the yeast, it's more I started working on bioenergy and biogas on a European project, but still I was very much interested to do that work. So I collaborated a colleague. He used to go to the woodlands, and we used to get some sort of the beetles or larvae that feed on the decaying wood and try to isolate some sort of the microorganisms and then see whether we can use it for the bioenergy or ethanol production. So we have isolated some yeast from beetles and also from soils. We reported new species even back in UK here. So it's that sort of the continue of the science, what we've done a bit more back in Hyderabad. It's still we continuing doing until this time. So once you isolate the microorganisms, you need to understand what the bug. It's like a new baby. You need to know what it is and what it can perform. So you need to clearly look into the colony morphology, what sort of the growth characteristics from the plate you can observe. Then you have a couple of hundreds of the plates. You know the differentiation. They are not the same. They perform different things. So you need to slowly try to read how they look onto the plate. What sort of the texture, what sort of the growth it can be. And then you have to take it to further. So there are several chemotaxonomic tests to identify the strains. For example, you have to do around 60 different strains for fermentation and as well as utilization of different sugars. So that will differentiate from the sugars. For example, in this case, if I'm looking all these sugars utilization, my idea is I want to isolate something methanol utilizing strain. So I have to see, is there any methanol positive strain for developing the strain? That's what the information is so crucial. So if you want to isolate some xylose utilizing, you have to check that whether it can be utilized xylose. If you're looking for methanol, you have to of the chemotaxonomics tests are very, very important. We take it very easy. We normally look into very few glucose or few carbon sources, but there are several. Now sources on the plate. So literally just you add your broth, culture broth, and then that will detect. So you can see here some positive, negative plates on that. So that will determine what sort of the carbon source it can be utilized. So you can use, this is more rapid, more advanced, because you can, instead of doing hundreds of tubes, here if you just if you put some broth, you get the results within few days and very, very accurate. So it's more easy to handle the automated sort of new characterization technique. So this will give you quite a lot of information. So why it's so crucial? So it's not only when you isolate some new species, new organism, then you need to prove that, how close it is the previous thing or how new it is. So you need to differentiate that thing. For example, here while I was working at Northwick, I have isolated this from the beetle gut. Then I have to show that how close this is with the previous strains, not only molecular biology, but also in terms of the characteristics of different sugar utilization. So that is very, very crucial to report the new species. So the first yeast species in the world, it's a pure yeast, isolated at Carlsberg laboratories in Denmark, where they produce this Carlsberg, the brew, the beer. But that's what it started actually, because at the time, a lot of contamination in the water, they couldn't understand before Pasteur discovered the pasteurization. At the time, it's very difficult to understand what's going on into the falling of the beer. So the scientist, I think Christian, he's the one first isolated pure yeast in 1883. So that revolutionized all the brewing industry in Europe. So that's what it started. It's like 175, 76 years ago. So the pure yeast was isolated, and that still continuation almost 175 years. Still they use the same beer, same sort of the bug, or it's called Carlsbergensis. They still use in several beer production in Europe. And now most of the breweries going into non-alcoholic, not only alcoholic, but it's also non-alcoholic. More than 50% of the brew nowadays more without any alcohol, but still the people taste that. So this is what it started in 1883. So not only they discovered the first pure yeast, but the techniques to isolate the pure yeast, but they also invented the pH meter. I think 1903 Carlsberg Laboratories, first time they started using pH scale. So that's very, very interesting. But beyond that, what they have done recently, if you go back, they have a cellar where from 1883, each bottle they store in their cellar. So they got a Guinness World Record. So there are thousands of bottles from different decades. So they gone back to the cellar, and then they have taken one bottle. That's the first batch they produced in 1883. So after so many years, they're able to isolate the yeast from that first bottle. And then they brewed another one. It's called Carlsberg Rebrew. That means it's fascinating you can still able to isolate an yeast from that old bottle after hundreds of years. So so what they've done in recently, they brewed again. They said that brewed with the original pure yeast from 1883. So they recreated the bottles. They done the rebrewing project for 3 years the same way they produced in 1883. So this is very, very fascinating. So those strains are very robust. It's viable for a long time. So that's what it started. You can see that he's been isolated by Christian Hansen in 1883. It's called Carlsbergensis, and they're given away. They haven't patented this strain. They're given that strain all over the Europe, and then so many breweries they use it. And now with the advanced genome technologies, you can identify what genes, what the transformation, what's the adaptations all these years while you're doing continuation of the brewing. So now the new genome technologies will tell you what sort of the genes in for the production of the base. So it's world's first pure cultural agar yeast. But not only that, if you use these advanced technologies, you can identify different yeast strains from different part of the world. The same way how we rapidly characterizing the COVID strains from all over the world. So you get the strains, then in a rapid way, you can identify what sort of the mutations happening in different regions of the world. So new gene techniques can allow us to rapidly to know more about the mutations, what's happening into different strains. The same way yeast, if you have different yeast from all over the world, now you can study how close they are and what's the diversity of them. So that's what the advanced genome sequencing will help us to understand. The genes are the adaptation of the genes, are the cutting of the genes, are the putting new metabolic pathways. We can do a lot of changes once you understand the genome of the particular bug. So as I said, at the time, we are looking for methanol utilizing strains, but I went everywhere. Even we went to Medaram forest to get the tree barks, and we have isolated yeast. So from fruits and barks and forest regions, hundreds of the strains we have isolated. For example, we have screened around 374 strains to test whether any of them can utilize the xylose. That's what can we produce from ethanol from the xylose. And also we have identified some sort of the enzyme production yeast strains from the forest bark strains. So we also look into the different diversity of the yeast that present in the forest region of that particular ecosystem. So here again, we have studied hundreds of yeast to know what they are they new or what they're doing in the forest economy, forest ecosystem. So why so important, those microorganisms? So for example, as I said, if you want to produce different molecules, different chemicals, like for example, here you can produce one of the molecule. I'm working more on the xylitol. I'm going to talk about it. So that you can produce some sort of the value-added chemicals and sweeteners and products from the lignocellulosic material, from renewable material. So it's very challenging, and then you need new microorganisms because it's very easy to isolate some strains that can utilize glucose as a pure sugar. But if you want to use for the biorefining, where you want to use the plant material, so you have to get for example, rice straw, or the wheat straw, or whatever the straws if you want to use, you have to get out of the sugars from the straws, and then during that time it produces quite a lot of inhibitors in the hydrolysates. So you need to have a different strains that can tolerate the inhibitors in the hydrolysis process. So we always look into new microorganisms that can tolerate different inhibitors in the sugars coming from the plant material. So it's very, very challenging. A lot need to be done. So it's always good to isolate some strains that can tolerate inhibitors in the plant material. So if you see the billions of tons of waste material all over the world, as I said, agriculture waste or the forest waste, tons and tons and million tons. Now in India, that is one of the problem. We're burning a lot of straws in the field. So it's not good for environment. We need to use those sort of waste material in a sustainable way to produce some chemicals and products from that sort of the ecosystem. So that's very important. Still, we need to look for more microorganisms. So basic microbiology, what I'm going to say, it's not about the vaccines, it's not about the fuels, it's not about the sweeteners, it's nothing, anything, any molecule, if you want to produce, then you need a good microorganism. So that is very, very basic thing. So end of the day, you have to have a skills to isolate good microorganisms from diverse ecosystem. So when I joined at IBUS, we initially started looking to xylitol. It's a huge market in terms of chewing gums and mints. It's around Cadbury's and Wrigley's in UK. They produce $40 billion worth of chewing gums, and that's useful, especially it will reduce the cavities. Streptococcus mutans, that's the organism that will cause the cavities, especially in the kids and school-going kids. They use a lot of sweets. They eat a lot of sweets. So if the xylitol is a sweetener, you can isolate from, normally produced in China from corn cobs through a chemical process. That's what they use in the chewing gums, and they eat the chewing gums that will reduce the cavity. So When I started looking into what sort of the market it is, 125,000 tons from the chemical process, it was in 2012. So no one produced biotech way. So while back in India, we started doing it 2000. And as I said, in 2012, we started at IBUS, the basic work based on this chapter, and no one produced. And I never thought, is this going to happen, the biotech process? Can we come up with a biotech process in my lifetime? I don't know. It's very challenging if you want to produce it from the xylose, especially from the agriculture material. Because you need to get the xylose from the plant material, either corn cobs or the sugarcane bagasse or the any sort of the xylose or plant material. But interestingly, after 20 years now, last year during pandemic 2020 December, one of the Chinese company, they claimed they produced first biotech process for xylitol. That is very interesting. But the technology taken from the States, USDA, they given the technology. They've been working from, I think, 1985, 35 years. I've been working 20 years, but they've been working for 35 years. I think they used a bacterial strain, recombinant strain, to produce the xylitol. That shows that, yes, it's viable with all sort of the challenges. It took like 35 years to get a biotech process viable on a commercial scale. So it's operating now. It's been 1 year. We don't know what sort of the financial situation with that company, but they claim that they're producing xylitol to biotech process commercially and they got into the market. So we still developing. We done up to pilot level. We spin out a company through university, and we going for next demonstration scale. Then that might be our strain is far better in terms of in vitro tolerance, and in the yeast we still working on it. So it's a continuous working. It's a continuous process to develop the strains. We've done quite a lot of microbiology and quite a lot of the gene knockout technologies to develop the strains for viable commercial production. So it's also, you have to understand where, if you want to go for a specific bug, what sort of the environment you have to see. Here, for example, I have started isolating the strains from gut of beetles. So some of the beetles from there back in Hyderabad, some photographs, and some of the beetles from UK and the larvae. So it's a lot of thing. So why we should go for gut of beetles? Why we should isolate the yeast from gut of beetles? So the reason behind that, the beetle larvae feed on the decaying wood, in any decaying wood, in a dead wood. That means they might have a system to hydrolyze the xylose, or they might have a system to degrade the xylon, or producing some enzymes in the wood to get the xylose out, not only glucose, but xylose. That is the idea at the time. So we looked into the xylose fermenting yeast from gut of Coleopters. Now, if you see, yes, that's correct, because the ecosystem, if you see here the gut segment of the beetle, and you can see the yeast also, what we can say, it's viable in the gut system. So it's a symbiosis. So that's what it's showing. So that means if you go for a very particular environment, you can isolate a particular strain. So here, that means the larvae of the beetle, they feed on the wood, and their gut system have some sort of microorganisms that can decay the wood material. So that's what interesting. So we can able to isolate some strains from the gut beetle. So we reported a novel species from the gut of beetle. So that's about the microbiology of it. So the interesting thing is you need to have a good microbe to produce any process or any chemicals. Then how are we going to translate that science into the biotech? Because as I said, it's a challenging thing. It's easy to do something in the shake flask, but if you go for scale up, there are several challenges. So you need to, so basically now you have a strain, you can understand what it is, what species it is by sequencing, because the sequencing costs down now. So 20 years back, it's very expensive, but now it's very cheap. So you can get it done, the primers, and get sequencing with so many institutes. Are the third-party laboratories. And then once you know the bug, you're going to do different basic studies. Can we use that for lignin utilization? Can we use that for xylose utilization? Can we use for glucose or different sugars in the materials? And what sort of the products we can produce out of it? So that's what the strain development you in the lab, and then you have to translate that into the production. So start with the shake flask and then go to the 1 liter bioreactors, and then you have to go for downstream processing too. So these technologies are more biotech based. That means the fermentation is so important to deliver the process or develop the process to deliver the product. Why it is so important? That means if we talk about the pandemic, then we have to produce billions of doses of the vaccine in a short period. So multinational companies, without knowing whether they're going to success or not, they invested like $400 to $500 billions of dollars, even though whether they know whether they're going to success or not, but they built the facilities, get going, right. Someone designing the strain at Oxford University, they're working on it for 10 years. So they are confident that they're going to get a strain. But meantime, to AstraZeneca or the other companies, they started building the facilities very rapidly to very advanced fermentation facilities. Once the strain is ready, just get going, produce so bulk and get going for the approvals. So this is what very challenging, because they know that the pandemic's going to come. So they working on the strains for 10 years, but the fermentation, we haven't been ready to produce such a bulk in a short period. So the facilities need to be ready. So it's very challenging. Now we understood how important the fermentation process, especially if you want to produce a product within a short period of time. And you also need to have understanding about the purification and downstream processing. So there are a lot of chemistry, microbiology, biochemistry, engineering, biochemical engineering, chemists. It's a lot of interdisciplinary work. So you can't just roll out based on only one molecular biology or microbiology. You have to have a interdisciplinary groups to deliver this sort of the challenging process within short period of time. So for example, here, this is very challenging. Why? So many universities, they do fantastic work. They do have a very good microorganisms, but they're not able to deliver a product to the society because the lack of infrastructure. So that is one limiting factor. So very few universities have that sort of the capacity of capability. Other big institutions like CCMB, very rich institutes, are very well supported with international support and national support with a mission or something. So they can capable of rolling out the technologies quickly because a lot of money invested. It's need a lot of money to do quite a good R&D piece of work. But saying that, I'm not saying the basic science is important. Basic science is so important, but also you need to have an opportunity to develop a process. So for example, here we have the shake flask, then we do have around 300 ml, very mini micro reactors where we can do a lot of fermentation optimization at 50 ml in a proper fermenter. Then you go for 1 liter, then you go for 10 liters, and then check it around up to 50 liters. And then once you've done that, if you have some sort of the confidence of the process, we do have a fermenter up to 350 liters. So we go up to that level, 200 liters or 300 liters. Then by the time you get to know how the bug behaving, what is the parameters of the fermentation, how we can improve by 1 liter to 10 liters, doing a lot of studies, and then go and do it around 200 and 300 liters. That will give you confidence that it works. Then you go for 1,000 liters somewhere. For example, in our case, we've done, I think, 4,000 or 5,000 fermentation at shake flask in 10 years. And then we gone to 1 liter. We might done maybe a couple of hundreds in the 1 liter. And then we done couple in the 50 liters, then 1 or 2 at 250 liters. But that results, and we replicated in Brussels up to 1,000 liters. So yes, that's what it is. It's a lot of difference between lab scale to the industrial scale. There is quite a lot of challenging. So biotechnologists need to understand how to run this and what sort of the parameters involved in it. So you need to know a lot of about the biotech process. Once you've done it 1,000 liters, it's easy to scale up. It's easy to go for 10,000 liters or 1 lakh liters. It's not a problem, because by the time you can understand a lot about the bug and a lot about the fermentation process. But this is the crucial thing. So pilot facilities really helps. I'm not saying not all the universities will have, but I think if they have a good bug up to 1 liter, 10 liters, with the infrastructures now available at third parties, so you can get going up to scale up. So it's not a problem. So as I said, we done quite a lot of fermentations up to 300 liters. So here you can see it's a steam in place thing, and quite a lot of sensors, quite a lot of automation involved in it. So It's one button. You can do all the sterilization within the vessel, and then you inoculate. Then you can adjust your parameters, and then quite a lot of controls. This is a software-based one, so it's easy as a touch screen, and then you just set everything, and it's get going automatically. But you need to monitor the process. So you can do a lot in terms of the shake flask, the fermenter, where we can control agitation, you can control the feeding, you can control the oxygen supply, you can dissolve the oxygen, you can control, you can feed intermittently, you can control the pH. There is a lot of things you can do. You can do even online sensors. We do a lot of sensors to monitor the process. So you can do more precisely if you have a proper fermentation fermenter to do that. So that's about the more aerobic, but we also done quite a lot of anaerobic work, part of the EU Agro Biogas, where they produce biogas. It's a very complex microbial process. So how it works, you get tons and tons of straws. They put it into a big digester, and then anaerobic bugs, they're going to produce this aerobic and anaerobic together, but it's like a complex process. First, a couple of hydrolyzing bugs, they produce acids and normal compounds from the raw material, like grass or the wheat straws or whatever the straws. And then you get the, like Clostridium, they hydrolyze the initial plant material, and then you get some sort of the sugars and acids, then that converted by archaea to the gas. And you take the gas, then you get the energy. Either you can produce heat or you can produce electricity by using burning the gas, and then that can be connected to the grid. So we worked quite a lot on a commercial biogas plants throughout the Europe. The thing is, the problem here is most run by chemists, and they can't understand this is a microbiology process. So how important it is. They do have around 10,000 commercial biogas plants, but most of them 50% efficiency because they can't understand the microbiology of it. So we always said that there is a room you can increase like 10 to 20% of the process because it's a microbial fermentation process, but 10 to 20%, if you're talking about 10,000 biogas plants, like you get, instead of getting in 100 megawatt, you're going to get around 110, 120 megawatt. 20 megawatt on that scale is important. So even by understanding and optimizing the bio process, you can improve the process on a larger scale a lot. So that's what we try to understand the microbiology of the biogas. So that's what it looks a plant. You're going to feed like hundreds of tons of the grass silage or wheat silage or the rice silage into that, along with the manures, and that will be get fermented in that with quite a lot of bugs. They do that process and they produce the gas. So what we done, this is the first large biogas plant in UK. It's around 4,000 cubic meter. They produce electricity and then the heat. We sent the DNA to the CCMB to do the characterization of the bugs. So at the time, we work with the CCMB collab. Still, we've been working for 15 years with India. I've been traveling regularly and working with this the institutions. So and then they done the characterization of that. They done the 6-ness RNA libraries, and they said as what sort of the things. Then we also work with the national genome facilities here to understand the microbiology of it. It's a very complex process, as I said, hundreds of the microorganisms. So here I able to isolate first yeast species from the biogas reactor, and I named it Shivaji, my previous mentor at CCMA, one of the renowned microbiologists. So just to honor him, then I put his name as a new species of the Cryptococcus Shivaji. So he was very, very inspirational. And you can also study the biofilms. That is very important in the anaerobic digester. So we studied something with Max Planck's Institute in Germany. Some of the PhD students, they worked, and I sent the strains. They done a lot of work on the biofilms of the yeast. So, and also we done with the Genome Analysis Center when they started here. How it started, it's called MRC. They started the Human Genome Project. The lady head of the Human Genome Project of the UK, she started an institute, and she called for different challenging things. They want to try to sequence using advanced sequencing things, one from animal, plant, and energy. So our project got selected on the energy side. So they done the 454 sequencing in 2010, a lot of work, and then we got quite a lot of information about the microorganisms of that particular area in anaerobic digestion. So you get the genomics, you can do the bioinformatics, and then you mine the data. You might have new genes in it, new microorganisms, and you can use novel enzymes from that system. So the genome sequencing helps a lot in terms of the process identification. And as I said, I do have quite a lot of India partnering awards to travel to India, collaborate. So we've been extensively traveling for the past 10 years to India, working with CCMB, work with IICT, ICRISAT. We do have a current project with ICRISAT. And even Gayatri, the project, they sponsored my PhD. They are still partner with us. They are our the industrial collaboration. So we've been working with India a lot. And I get an opportunity to go to the college and deliver a couple of lectures with my colleagues at CDC. That's very interesting. So it's been like a bridging the lot of collaboration with India. I have done a lot because now we're out of the Brexit, so we're looking for more partners in India and Canada and States. So there's a lot happening with India now in terms of UK-India collaboration on a larger scale. I'm going to talk about that in a minute. So the process control, we developed a pilot plant to understand the microbiology of it. So we done a lot of automation on the pilot plant. Then we sequenced a lot of the samples, gone to the genome center. We know what sort of the microorganisms. We are refining a lot for VFA production from the reactors. So to understand volatile fatty acid utilizing microorganisms from anaerobic digesters. So nowadays, more early warning system, more sensors availability there, more rapid technologies. For example, NIRS, you don't need to take the sample. You have a probe, you put it onto the glass, it will measure what's going on inside the fermenters. They use for the glucose monitoring in pharmaceuticals. So a lot of the sensors now available, a lot of automation available, quite a lot of rapid developments for the basic fermenters. So they in terms of the microbiology, in terms of the sensors, in terms of the monitoring technologies, quite a lot happening. New protocols, new methods, non-disruptive methods. So it's more fast, more rapid, a lot. The next level, we try to, for example, if we go here, we do have the system. We haven't gone to the pilot plant. It's automatically run, all the feeding, everything from the computer. So you can see what's going on your computer. And with some sort of the hardware and software, it's automatically fed, automatically collected the data and feed into the computer without going any manual intervention. That means you don't need to go to the pilot plant. But if you have a hardware and software, you can monitor everything. Literally, you can monitor from India, from on your phone. That sort of the technology is now available. So for example, sitting here from my computer, we can control some fermenter back in India. So that's sort of the advancements there now. So you literally can do wonders with advance the technology of the process and on phones and everything. So that is very interesting. So this is first sort of automation where we monitored more remotely. Most of the time we show to the people on the monitor what's going on in the plant. No need to take them to the plant. So that is about monitoring technologies, process control technologies. So here we do have a pilot plant where a lot we've done on a larger scale. So you can see this is like a double-decker bus. You can biomass, you can like wet grass or wet biomass, if you can dry on this like a couple of tons in a day. So quite a lot of facilities you need to roll over. So we do have a primary processing labs. We do have a secondary processing labs. We have fermentation capabilities to do up to pilot, one of the best pilot facility in UK and even in Europe. So how it works, we do a lot of work with the industry because very little with the students or the basic research. Most of the projects we work with Welsh industries. It's like a Telangana, Wales. We located in Wales, and then we work with quite a lot of Welsh companies. For 10 years, we working with hundreds of companies to different things. For example, you have you have some material, you want to do some sort of the work, we get it here. Sometimes you may have the material, but you don't have the fermentation capability, so they come to us, we do the fermentations. Sometimes they may have the fermentation capability, but they may not have the downstream processing purification, so we can do it. We have pasteurizers, we have a lot of centrifuges at pilot scale, we lot of what we can say purification membrane systems we have to take out the product. So here, once you understand, sometimes when you work in the lab, you can't understand the complexity of it. For example, You work on 1 gram, 2 grams, but when you go for large scale, how are you going to feed that? How are you going to feed tons and tons of material? How are you going to take out? How are you going to take out the material? How are you going to purify the material? Like thousands of liters or 10,000s of liters. So most of the processes, if they want to go for demonstration and commercialization, then you need to have a solid data that works at the pilot level. So pilot plants are so crucial. So it's an fortunate to get a work in this sort of the facilities here. It's funded by the government. I think they invested 40, 100 millions. Maybe in terms of our money, it's like maybe 800 to 1,000 crores in 10 years time. It's a huge money. But that's what it is. If you want to really develop a technology, then you have to invest. That's what happened at Oxford. It's easily they developed the technology, they given to the AstraZeneca because they've been investing on the medical biotechnology for more than 10 years. So I learned a technique called process optimization from a mechanical engineering professor while traveling on a train, but we rapidly brought into the biotechnology. This is I learned from Shin Taguchi, the Taguchi-san, and as well as the Professor Rajesham, a mechanical engineering professor from Malaysia. So I learned this and we brought it to the biotechnology. And now in all over the world, so many people, they're using this process optimization, more statistics based. It started at Rothamsted Research, where I used to work in 1920s, R.A. Fisher. And then C.R. Rao from India, he came and worked with him at Cambridge University, and he developed this orthogonal process. So Indian Statistical Institute really helped a lot to roll out this Taguchi methods for the industry. So all this started at universities. As I said, the institutes at Rothamsted Research in 1920s by R.A. Fisher, F. Ratio, ANOVA, and all. And he's the one first demonstrated design of experiment to understand the different variables for the crop yield. Then it went on to the industries, and it's a big thing. You can save a lot of money. You can understand the value of it when you can't understand when you do it on a 50 ml or 100 ml. But if you want to do it like 250 liter fermentation, then you need to be ready with all the processing, because you can't run hundreds of experiments at 250 liters, like how you run at 250 ml. So this is very, very important tool on a larger scale. So as I said, we're working with India, with Gayatri Bioorganics to refine the sugarcane bagasse to xylitol, and also for AD and also for VFS production. We're working with the IACT, and we pulled a lot of companies back in UK, Membranology for downstream processing, Mandrakem for engineering side, and it's funded by the Innovate UK, or BBSRC for us, and the DBT funded the IACT and Gayatri. So we're working on this for 3 years now. We come up quite a lot of value products up to pilot level. So maybe Gayatri can take up some process to test at industrial level. So that's what it's boiled on. It's been for 3 years with India. We're working on this project with several companies back in here and back in India. So for example, how it works. So we don't have a sugarcane fields in the UK. So we don't have sugarcane bagasse. Literally, they shipped 4 tons, around 400 or 500 bags of sugarcane bagasse in a container from India to UK. We got it during the pandemic time. We processed all those things, hydrolyzed it. So you can see thousands of liters of hydrolyzed. Now we started fermentations up to 1 to 2 liters. We're going to go next year for up to whatever the scale up. And then we're going to see. So a lot of results going to come out of this process up to pilot level within the next few months. So that's what it works. You need a lot of facilities to roll out the technologies, to get a confidence in the companies that, okay, these technologies can be developed. But it takes a lot of time to understand the process. As I said, we can't do in a one go microbiology. So you need to learn slowly, start with the isolations, then learning a bit of bioinformatics, learning a bit of sequencing, learning a bit of fermentation. Then you take it layer by layer by layer. It takes like to master something. To get to this stage, it took 20 years for me. So it's a lot of work and a lot of things. So as I said, that's what it started at Hyderabad. Now we ending at Aberystwyth, 10 minutes to go. So I'll speak few things here. So recently, Wales collaborating with the Telangana government. It's not about India, UK. I'm talking about a region called Wales, like in Telangana. So Telangana government, higher education, signed an MOU with Welsh government to cooperate universities to universities, Welsh university, 8 universities to Telangana University. So that signed MOU in last December 2020. So first initiation, they sponsored, I think they called for 16 students, Telangana students, especially from Osmania Kakati University, to come and work, come and study masters here at Welsh universities. So they get around 5,000 rebate on their fees if they get selected. So that's what the 16 students funded between the universities, between the government, that is funded by the UK government to promote students come to Wales from Telangana. So that is just an initiation. So, but there are beyond that. Now they're changing the syllabus, especially for arts and humanities for Osmania University and Kakati University. That started, and the next stage we might try to revive a bit of microbiology or biotechnology syllabus at Telangana universities. So there is a global head in Wales. So it's called a global program, Welsh program. So on that note of MOU, that I think first humanities and going to be started already, I think, with Osmania University and Kakati University. I'm not sure who's going to involve in that. But they asked me to involve in some sort of the science program things to do some sort of the curriculum change or whatever. So there's a lot going on, a lot of changes. So we need to really update. I think some point, it's not a criticism, but maybe the syllabus, what I studied in 1993, 95, the same syllabus might be continuing at the moment after 25 years. So we need to adapt a bit. We need to change a little. I'm not saying that we need to change drastically, but I think some change need to be, it's a given a chance. So we might work more collaboration. I don't know in future, but that's what it is at the moment. So I got invited by recently Welsh First Minister, and also Indian High Commission was there. So we have a roundtable discussion to see how we can work more with India, not only with India, especially with Telangana. So it's the first one is to work with, to collaborate more with higher education institutions, but not limited to universities. They might working with institutes. They asked me more about Hyderabad Institute. So I introduce all those institutes to work with maybe companies, businesses too. So startups, whatever it may be. So it might be a lot going to be happening, and then a lot of people going to involve. Maybe we might come next year, or some teams from UK universities might visit Hyderabad institutions, and then there might be some more dialogue or more collaboration going to happen. So at that note, I would like to finish. As I said, so it's in terms of welfare of the microbes. As I said, it's not just medical or therapeutics. You can use it into medical. You can use it in industry sectors a lot. As I said, the yeast, the worth of the yeast biotechnology is around 4,000. What's that? I don't know. I forgot. 43,000 crores per annum. So a lot of opportunities to develop new process. It's not just we don't need to focus on what the available process, but always there is a novel ingredient, novel processes, because of the health complexity, more diseases. So we need to ready with more nutritious products, more refining of the product. So a lot of opportunities to work with. So there are more challenges and also more opportunities. So thank you very much. So this is a nice place to live. Maybe I got a new yeast. I might name after Aber. Maybe, I don't know. Let's see.
Thank you.
Thank you very much.
Thank you, Srinivas sir. And now we'll give time for queries, sir. Students, you may ask your doubts. You can unmute yourself and ask your queries. Or you can post in chat box also.
I think I would
Sir,
like
one
to
of the students.
Dr. Suresh, Dr. Challa Suresh. So one of the initiation is that when they came to the delegation from IITs and the Indian High Commission in Cardiff last week or 2 weeks ago on Diwali reception, they funded they exhibit called Namami Ganga project. I think you might be knowing, I'm not sure, but that's what it is. So that is a big initiation. They're looking for technologies to clean up Ganga for several things, not only water purification or the pollution from the factories, but also floodgating and sustainable agriculture. So they funded, I think, past 10 years on a flagship 374 projects for around $4 billion. So I think water purification, for example, lead, if it is there in the pollution, then how that going to be clarified or clear, and what is the impact on the as that is a wonderful lecture what you given in terms of environmental pollution. But so they are looking always new projects. It's called Namami Ganga. There is a website. They're always asking for new processes, new technologies, new research with industries. They may not fund the basic university projects, but a pilot, or if industries collaborated, then they're going to give the money. So there is a lot of money available in India, not only UK, but it's how this can be approached. So I would like to thank you, everyone, organizing committee, and then my teachers and the students.
Srinivas, wait a minute.
Yeah, sure. I'll be here.
One student, he posted a question.
Yeah.
How can we expect the same concentration of byproduct or metabolite from shake flask level to pilot scale and demo scale? What are the parameters that would affect the concentration of the product?
Yeah. So the dynamics is completely different between shake flask to the fermenter. For example, it's more aeration. You get more oxygen diffusion in the fermenter because of your spinning, and you can supply the air or oxygen to the reactor. So the process might be faster and process might be better. So those two are completely different. What you get in the shake flask, that may not exactly you get in the 1 liter or 10 liters. So the dynamics will be changed. That's what you need to understand what's happening. The process might be better or worse, but you have a scope to improve the process once you understand the aeration of that bug, how much it's growing in the, even for example, in a shake flask, there are few things. If you take a flask, if you put 100 ml, and then, for example, if you put 150 ml, and if you slowly shake it, you will have a microaerobic condition. Whereas if you put 50 ml in a flask, and if you do rapid aeration, rapid agitation, 250, you giving more oxygen to the cells. So it will more aerobic. So there are some papers how you can do a little bit of aeration in the shake flask. That means you take a lower volume, 50 ml, and then high shaking, give you more aeration. Whereas if you take 100 ml, medium agitation, give you moderate aeration. If you take 150 ml and then do a slow shaking, that will give you a micro aerobic condition. So you can create without supplying the aeration to the flask, but still you can do a study on it. That's what you need to understand. Then you have to do a little bit of 1 liter. Then you get to know how the aeration need to be checked, because you can use the same nutrients, but I think this aeration will help. Yeah. Is that clear? I don't know.
Elaborate the impact of second metabolites on expression in yeast fermentation. This is from G. Praveen Kumar, sir.
Sorry, come again, Lata. What it is?
Yeah, I'll repeat it. Could you please elaborate the impact of secondary metabolites on expression in yeast fermentation?
Secondary metabolite on yeast fermentation. Okay, so what sort of the secondary metabolites? Yeah. What sort of the secondary metabolites?
Yeah.
Is it using a expression systems, or it's just a normal secondary metabolites? Is it expression systems, molecular systems, or the general microbe you're talking about?
Expression system, it seems.
Oh, okay. Pichia pastoris expression systems. Okay. Okay. So if we talk about that, sorry, is it okay? Can I speak now? Yeah. So the Pichia pastoris, I think that is the first metallotropic expression system available in the market by, I think, MCT. Microbiological expression technology, or the research expression technology. That is the company, they develop this technology. How it works, I think there are 5,000 different therapeutics already expressed in pitch Aberystwyth. Very robust system, methanol induction, nice, everything works. The only problem with that, once you take the technology, they license you for 1 year. You can use in your laboratory. 1 year, you can use it freely. Once you buy the system for 1 lakh or 2 lakh, you can express whatever the gene you are interested in. You can produce some product. You can check everything. So they will give you an year time to do all the basic research. But once you got there, you got your gene express, some insulin, whatever it may be, you like it, you want to produce the metabolite using that particular system, what they will say, the next step is that if you want to scale it up to 1,000 or industrial scale, they will ask you the royalty out of it. The royalty might be 1 to 5%. That means if you sell 100 doses or 200 doses, then you have to pay royalty. Initially, you don't feel the pinch, but when you talk like Shanta, when you are producing billions of doses, then you have to pay billions on the royalty. So that is what we want to try to develop a new expression system. But Pichia pastoris one is the very robust and in vitrogen cells. So many companies, they sell the technology. But as I said, the kits, you get it for 1 year. And then after that, if you want to use it on a commercial scale, still you can use it, but you need to pay the royalty. And that is very robust system. As I said, there are 400 or 500 different molecules expressed, and a lot of information is there on the net about the Pichia pastoris system.
Thank you, Srinivas sir, for your interesting and informative talk. Sir, really, you have taken us to the fascinating world of microbes. For some time, we thought that we are in Europe. Really, we surprised to know the facts about microbes and the challenges regarding biorefining, scaling up the process, Taguchi methods. Thank you very much, sir. People like you are hard to find, sir. You are one living example, true motivation for our students. You started your journey from our college and now you are one of the prominent personalities in Aberystwyth, UK. We are proud to have an alumni like you, sir. Always you are trying to inculcate values and research activity in students whenever you get a chance. Really, thank you very much, sir, for sharing your knowledge with us. Thank you very much, sir. Now I invite Dr. Srilekha, madam, to present vote of thanks. Madam, over to you.
Good afternoon, one and all. I, Dr. Srilekha, on behalf of Department of Biotechnology and Management of Chaitanya University, take it as a privilege to propose vote of thanks for today's webinar. First and foremost, I extend my sincere thanks to Mr. T. Vikram Reddy, sir, for taking time out of his busy schedule to be with us here today and for gracing this occasion with his presence. I would like to express my profound gratitude to Professor T. Krishnamachari Garu for his blessings and motivation. I take this opportunity to express my profuse gratitude and sincere thanks to Professor M. Ravinder sir for your support and encouragement. I would also like to take this opportunity to express my deep sense of regards to Professor Anuradha ma'am for being here with us and for her support. I would also like to thank our speakers, Dr. C. Suresh sir and Srinivas sir, for enlightening and enriching us with their knowledge. Both the talks were very informative and interesting and has revealed very interesting facts. I am pretty sure that this knowledge will definitely help the participants in future. Thank you all once again. And now I have some announcement to be made. We have student poster presentations today in the afternoon at 2 o'clock in 4th floor seminar hall. And tomorrow we will be having online poster and paper presentations at 2 o'clock in the afternoon. We will meet again tomorrow at 10 o'clock. Have a nice day ahead. Thank you all once again.
Thank you, madam. Thank you very much.
Thank you, sir.
Thank you, Rajendra sir, for everyone, organizing committee, Lata and everyone. And I wish you all the best for the students. And if you need anything, please contact Lata, through madam, and we can chat about it. No problem.
Yeah.
And I will be in touch with you, sir, Suresh Garu.
Thank you, sir. Thank you, one and all.
Thank you.
Thank you, sir.
Hello. Hello. Hi.